Hi guys, Miss Francis here to talk about the stages of meiosis. My suggestion, if you have not yet watched the vocabulary review video, do that first and then watch this video. The understanding of those vocabulary words is important to understanding the stages of meiosis. Now, throughout the stages of meiosis, there are 3 vocabulary words that we haven't been introduced to yet: tetrad, synapsis, and chiasmata. So let's jump in. We already know that meiosis reduces chromosome number from diploid to haploid, where meiosis is preceded by the replication of the chromosomes. That happens during the S phase of interphase. During meiosis, there are 2 consecutive cell divisions. There's going to be meiosis 1 and there's going to be meiosis 2. Now, the daughter cells at the end of meiosis 1 and at the end of meiosis 2 will both have half of the number of chromosomes that we started out with. So let's take a closer look. If you look at this image, do you see how we've got interphase that's going to occur before meiosis actually takes place? What that does is replicates my chromosomes, where I've got 1 chromosome from dad and my 2nd chromosome from mom. During the end of meiosis, these homologous chromosomes are going to separate. So now I end up with daughter cells that each have 1 chromosome in them. These are replicated chromosomes, but there's still only 1 in this cell and 1 chromosome in this cell. So we started out with 2 and now we ended up with 1. We ended up with half the number of chromosomes in the daughter cells. Now look at the daughter cells at the end of meiosis 2. Do you notice how all of these cells also only have 1 chromosome? In meiosis 2, our sister chromatids are going to separate and go to either end of the pole and then be pinched off and form its own cell, be a chromosome in its own cell. But once again, these cells only have 1 chromosome. Now remember, we started out with 2 chromosomes. So once again, we have half the number of chromosomes present. Before meiosis occurs, interphase occurs. And we know this. It's part of the cell cycle. During interphase, the S phase of interphase specifically, chromosomes are replicated and I get sister chromatids. So now starts the first part of meiosis. You've got prophase 1, where our chromosomes condense, or you can technically say your chromatin condenses into chromatids, which then condense into chromosomes. You've got that supercoiling going on. Now you've got homologous pairs. So here you've got the chromosome from dad that's replicated, and here you've got the chromosome from mom that's replicated. Those homologous chromosomes pair up at what's called a synapsis and form tetrads. Crossing over is what's going to occur at prophase 1. Crossing over is really important. I like to think of it as the chromosomes are kind of playing footsie. During crossing over, chromosomes can share their DNA. They can give some of their DNA to 1 chromosome and take DNA from another chromosome. The location at which these chromosomes, the homologous pairs, share their DNA is called chiasmata. Furthermore, you have your spindles forming. What I'd like to do is trace a cell through this process. So this cell's diploid number is 4, meaning I have 4 chromosomes. So I have 1 chromosome, 2 chromosome, 3 chromosome, and 4 chromosomes, where they are matched up into their tetrads. In other words, the homologous pairs have come together. So in this one cell, I have 4 chromosomes, but I've got 8 sister chromatids, right? 1, 2, 3, 4, 5, 6, 7, 8. Now let's continue on with metaphase 1. So now those tetrads, those pairs of homologous chromosomes, line up down the middle, down the equator of the cell. And we still have our 2n number. We still have 4 chromosomes present, right? 1, 2, 3, 4. Moving on to anaphase 1, well, now our homologous chromosomes separate, but our sister chromatids remain attached at the centromere. So here's my first homologous chromosome, here's my second homologous chromosome, here's my third, and here's my fourth. I still have 8 sister chromatids. I have 1, 2, 3, 4, 5, 6, 7, 8. So at this point, I still have my diploid number of chromosomes in my cell. But what happens next in telophase is the splitting of the cell. So the homologous chromosomes move toward the poles, and now I have 2 haploid cells that are gonna form. So now my number of cells is 2, but I've only got 1, 2 chromosomes in each cell. Those chromosomes have 4 sister chromatids, 1, 2, 3, 4, 1, 2, 3, 4. But now I only have n number of chromosomes. I have the haploid number of chromosomes in each one of those cells. So this is the end of the first division. And like I said, at the end of each division, the daughter cells are going to have half the number, or the haploid number, of chromosomes as what we started out with. So remember, we started out with a cell that had 8 chromosomes. Now we've got cells that only have 4 chromosomes. Moving on to the second division, you've got prophase 2. The spindle forms. There's no additional crossing over. Metaphase 2, now those sister chromatids are lining up down the middle. So in metaphase 1, you had the tetrads lining up. Now you have the sister chromatids lining up. And then in anaphase 1, it's the sister chromatids that separate. So in anaphase 2, it's these sister chromatids that are going to move to either end of our cell. So how is it different from anaphase 1? Well, in anaphase 1, it's those homologous chromosomes, or the tetrads, that are separating, whereas in anaphase 2, you've got your sister chromatids separating. So if we continue, we move on to telophase 2. Now my separated sister chromatids arrive at the poles, my nuclei reform, and the cytokinesis occurs, which separates the cytoplasm. So now I end up with 4 cells total. 1, 2, 3, 4. Where within each cell, I have 2 chromosomes. 1, 2, 1, 2, 1, 2, 1, 2. Furthermore, I only have 2 sister chromatids. Whoa. Uh-oh. Oh, God. I don't know what I just did. I only have 2 sister chromatids in each cell. 1, 2, 1, 2, 1, 2, 1, 2. So my cell is still haploid. I still have half the number of chromosomes in each cell as I started out with. Those are the stages in meiosis. If you're confused about what happens at each stage, make sure that you come see me or Miss Cunningham. My recommendation is to write out the stages, draw them out, and label each stage, and label what important things happen during each stage.